Hello, I'm Sally Kornbluth, president of MIT, and I'm thrilled to welcome you to my podcast, Curiosity Unbounded. This podcast is your chance to join me as I talk with pioneers who are pushing the frontiers of knowledge and inventing real-world solutions for a better future. Today, my guest is Sebastian Lourido. Sebastian is an associate professor of biology here at MIT and a member of the Whitehead Institute for Biomedical Research. Sebastian studies human pathogens and uncovers the genetic pathways that allow parasites like Toxoplasma gondii to survive and replicate inside human cells. His work could pave the way for new treatments for Toxoplasmosis, which affects roughly 60 million people worldwide, and inspire drug discoveries for diseases caused by similar parasites. Sebastian, welcome to the show.
Thank you, Sally. What a delightful opportunity to share our research with you today.
Fantastic. So you've said that your work grows out of a fascination with how different organisms coexist. What does that mean in the context of parasites? And how does it shape the questions that you ask in the lab?
Yeah, I think I have from an early age and some of the kind of early exposure in undergrad to different questions in biology, a fascination with how different organisms clash, right? Different natural histories with very, very distinct adaptations, all of a sudden coming together as in a process of an infection and using different tools to adapt to very different niches to what we typically think about as free-living organisms out in the wild. And so for me, I think there was an intrinsic fascination in that clash, but then furthermore, an intrinsic fascination with being able to observe that under the microscope. Mm-hmm. And because parasites are among the largest pathogens that infect humans, we can readily observe the process of infection, of parasitism, of intracellular replication inside of human cells under the microscope. And that really just captured my imagination. Moreover, in this class of organisms that we've decided to study, there's so little known about their fundamental biology. They've led a very distinct natural history from the branch of life that gave rise to animals and gave rise to fungi. And so many of the lessons at a molecular level that we've gleaned from studying those other organisms are not really applicable to these organisms that have followed a very distinct evolutionary history. And so there was so much unknown. I kept asking professors and colleagues what we knew about how this process happened and how that process happened, how parasites enter cells, how they draw nutrients from those cells, how they decide between when to replicate and when not to. And all of those questions seemed vague and unanswered. And so it just seemed like a really incredible area to go into research in, and also the impact that research could have and the lives of people suffering from infectious diseases just seemed like a wonderful thing to devote myself to.
No, that's great. I mean, this is far from the, public conception of like parasites in horror movies, although, the word parasite, I guess, implies an interaction that may not be great for the host.
Yes.
Unlike when we talk about, symbiosis, things both benefiting mutually together. This is, as you said, the parasite sort of hijacking, if you will, host functions. And I assume that made it harder to study as well, because a lot of the biology is not just intrinsic to the parasite, it's what the parasite will elicit from the host.
That's absolutely correct. I mean, I think that we can take that at several layers. Many of the parasites that we're interested in only survive, only replicate inside of a host, right? And that distinguishes them from maybe some of the bacterial pathogens that can be, in some cases, propagated in vitro in a flask. And so that has an inherent challenge that lays obstacles in terms of genetic manipulation, understanding of the molecular biology and biochemistry, all of the things that have been much more accessible in free-living organisms are indeed challenged by these sort of natural constraints of the system.
Yeah, that's interesting. I think we don't think about that probably as much as we used to now that there have been so many innovations in molecular and cellular biology, but a lot of discoveries were using systems that were predicated on the fact that they were experimentally tractable.
Yes.
And so, dealing with something that's out of view, hidden inside the host,
Yeah.
that you have no way to propagate in vitro, could be very, very challenging.
It's a major challenge. And certainly some of the major advances have been, how do we bring that as close to an in vitro system as possible?
Right.
So in our lab, for instance, in the case of Toxoplasma, we can maintain cultures of that parasite by infecting human cells in vitro and propagating it that way.
Yes. Yes.
And certainly one of the advantages of Toxoplasma is that it is relatively long-lived outside of those host cells. Even though it doesn't replicate, we can still manipulate it genetically. We can still change and observe elements of its physiology before putting them back into culture in those host cells, in those human cells, in order to allow them to replicate. So there are still other barriers for the study of other organisms. And there too, I think there's this advantage of relying on those particularly closely related representatives that are more tractable.
Right.
And that's been the case for Toxoplasma in this broader group of organisms called the apicomplexans, which of course include many other human parasites, such as the ones that cause malaria, some that cause diarrheas, and particularly intractable diarrheas in young children in developing countries, belonging to the genus Cryptosporidium. And then other kind of emerging diseases actually right in our backyard in New England, such as babesiosis, which is a tick-transmitted relative of Toxoplasma. So many, many diseases that can be, in some subtle ways, modeled back in the biology of Toxoplasma, which also motivates our research.
That's very interesting. And, sadly, we're going to see more of those parasitic infections arising with climate change, et cetera, and with close juxtaposition of farm animals, agriculture, et cetera, to humans in different areas. So let's talk a little bit about toxoplasmosis. For listeners who may not be familiar with it, tell us what it is, what effects it can have. My most recent reading of toxoplasmosis was probably 33 years ago, when I was pregnant with my first child and was worried about toxoplasmosis. So maybe you can tell us about it.
And what did they tell you then?
Not to change the cat litter.
Yes,
and
exactly.
That was great, because I had my husband always changing the cat litter.
But it probably stayed that way for a few
forever,
years, just to be safe.
As long as we had cats. Anyway.
Yeah, so the reason that you were told that is because Toxoplasma has a special interaction with the cat. When a cat becomes infected with Toxoplasma, which occurs through the consumption of maybe a mouse that was itself infected with the parasite, the cat can allow for the sexual development of Toxoplasma. So there are specific parasites that can live by replicating asexually, sort of just dividing and making more copies of themselves, or sexually by making male and female sexual stages that then fuse together to produce diversity and through sexual recombination. And so in the case of Toxoplasma, that sexual recombination only occurs in felines.
Interesting.
And the product of that sexual recombination are these very environmentally resistant stages that are exquisitely infectious to us. Probably just a handful of these stages coming into contact with our food and with our water are sufficient to infect a human being. And then in the context of a human being, what we would consider an intermediate host, if the cats are the definitive host, these parasites can go from our intestines and disseminate throughout our bodies. In most instances, our immune systems are really good at keeping those parasites under control, although they never clear them, actually.
Interesting.
So it's an interesting fact that of all the people who have become exposed to Toxoplasma, probably most of them carry the parasite chronically inside their tissues. And that's
so is that a problem for a subsequent pregnancy then?
It wouldn't be, because you already have the immune responses.
Gotcha.
And only the acute stages that are developing during that initial encounter are capable of traversing the placental barrier and infecting the developing fetus.
I see.
That's the real risk.
I see.
The other risk, which I think was really prominent during the AIDS crisis, was that reactivation of infection in those immunocompromised individuals could now allow the parasites to replicate unrestricted and damage all of those tissues where they were previously found.
Where do they hide out?
They hide out in muscle and in the brain.
Oh,
in
interesting.
neurons. And so, of course, that's a particularly susceptible place to have inflammation, to have tissue damage that can cause very severe diseases and even death.
So when they reemerge, do they begin replication in vivo again? Is that the deal?
Correct.
I see.
And in fact, a lot of our early work in my lab at Whitehead had to do with us understanding that cycle between them replicating and then bursting out of those cells.
I see.
And that bursting out will damage, of course, the cells where they were previously replicating and cause tissue damage that alters a lot of structures in the infected organism, right? It's no good to have your neurons dying off, having immune cells infiltrating those areas, causing more inflammation, more damage. That process is sort of predicated on parasite replication and host lysis.
You know, it's interesting. You know, I think there's a lot about this that we don't understand. So, for example, recently you probably saw the study that shingles vaccine is protective against dementia, presumably because the virus is lurking in your neurons for decades and decades and reemerges. I wonder if there's any possibility that, you know, Toxoplasma caused some late-stage neurodegenerative disorders.
This is an active area of study, and I think a fascinating direction, Sally. I think we don't understand what the long-term consequences of that interaction between the parasite and the host is. And it is one of my future aims to try to unravel the kind of complex relationships that are carried out over the lifetime of a chronic infection. I think we've made a lot of headway in understanding some of the genetic circuits that mediate parasite differentiation from that acute state where they're replicating rapidly to the chronic state where they can lay dormant inside of our tissues. We've understood now what the master regulators of that process are at a genetic level. One of our kind of big breakthroughs was uncovering the basic elements of that circuit. But what are the long-term consequences of having those chronic stages there, replicating slowly, drawing nutrients from our neurons, and ultimately causing low levels of inflammatory responses that we really don't sufficiently understand? How is that changing brain physiology over the long term? I think that is a really promising future avenue. And there are, in fact, associations at epidemiological level between Toxoplasma seropositivity, which would be the production of antibodies by people who've previously encountered Toxoplasma, and their prognosis in terms of neurodegeneration.
No, very interesting. That's really interesting. So maybe I was right to never change the cat litter again. So just to clarify for the audience, though, cooking meat thoroughly, taking care when cleaning the cat's litter box can reduce the risk. But what kind of treatments are available, especially for people who are immunocompromised?
So those individuals will have to be, of course, I should preface this with saying that I am not a physician, right? And that any sort of treatment should be in conversation with one's physician. But oftentimes, people who are severely immunocompromised are maintained on therapies that kill the actively replicating parasites. But it is true that right now, we have no therapies that are providing complete clearance of the parasites. And those chronic stages seem to survive every antiparasitic drug that we have right now against them. So when we think about Toxoplasma, we think about the suppression of tissue damage by killing off those acute stages, but not really a cure.
And there might be a way to induce the replicative phase and then target, you know, with antiparasitics.
I think that's a brilliant idea, and certainly one of the reasons why we continue to try to unravel that circuitry that dictates differentiation. And so we know, in fact, that some of the gene regulatory elements that we've found are required for parasites to enter the chronic state are also required for their maintenance of that chronic state.
I see.
So if we could figure out ways of targeting those, we would definitely be able to transform the parasites into stages that we can treat actively and cure.
Yeah, very interesting. So does Toxoplasma impact other species as well besides humans in terms of pathology?
Yes, and this is an interesting element of Toxoplasma compared to maybe some of its relative parasites,
Mm-hmm.
that it seems to be very poorly specific.
Interesting.
Seems to have cracked the code of infecting any warm-blooded animal.
Mm.
And we find the same types of Toxoplasma that humans can get. We find it in chickens, which are also warm-blooded, in mice, in rodents. We've previously documented the presence of these in alpacas. So
But they're not contagious from those species
only
humans.
when the meat is consumed,
if
right?
it's not well cooked.
If it's not well cooked, correct.
Oh, interesting. So does this actually serve, you mentioned a little bit about this serving as a model for other parasitic infections. Can you talk a little bit about that, how you would think about elaborating this work to other pathogens?
Yeah, I think that there are some conserved elements that are common to all of these parasites in the apicomplexan group. And that includes the need to enter cells. There are both differences and commonalities in some of the machinery that's required for that entry process. And we've done some work in discovering some of those conserved elements. We've also been able to identify common elements of their cell biology. After all, they all share common ancestry,
Right.
and they essentially adapted from the same free-living ancestors as they undertook this sort of path of parasitism. And so many of those elements are conserved, how they regulate their cell cycle, how they structure their cells, and the basic machinery that they use in order to start manipulating host cells. And then on the fringes of those interactions, maybe some of the specific receptors that they use to attach to host cells, or the proteins that they're secreting into host cells to manipulate them, those tend to be quite species specific, because of course they are under extraordinary selective pressure.
Right. Because they have to coexist.
The hosts
with the
evolve
host.
to try to suppress them. Yes.
Right. It's interesting to think about how these parasites originally evolved.
Yeah, it's I think a fascinating question.
Yeah.
There's some clues, and so I've for a long time, I think since I've started working in Toxoplasma, I've been interested in that question, as many of my colleagues. And there are some free-living relatives that you could find maybe swimming around in seawater, maybe in association with corals, including some organisms that have only very recently been discovered, as recent as 2010. Those organisms seem to have this sort of facultative association with other cells.
Yes.
And there's in particular in some of these free-living organisms the propensity to attach to other single-celled organisms,
but not get
and
internalized.
Suck out their insides.
Oh, that's amazing.
As a source of food. We call this cellular vampirism.
Yeah, I love it.
And so one can speculate, of course, it's sort of difficult to recapitulate all of this biology, but one can speculate that cellular vampirism all of a sudden went too far, and established a much more intimate association between the vampire and its host, if you will.
So I should know this, but I don't. So how easy is it to manipulate the genome of these parasites?
It differs between the different members of the group. In the case of Toxoplasma, I think we've been able to develop some of the best tools out of the entire group of apicomplexan parasites. And that's what makes it such a robust model for molecular biology, biochemistry, and examination of essentially some of the fundamental elements of how these parasites work. We've been able to use the advent of CRISPR-based genome editing to perform genome-wide CRISPR screens that have individually disrupting every single one of the 8,000 genes in Toxoplasma, and ask sort of what is required for infection of human cells, or for survival of parasites under different condition, for the acute infection that causes so much pathology. So that's been really, really transformative. And it has served also as a framework for work from colleagues that has attempted to do sort of large screens in malaria parasites, for instance. And it's been really fascinating to be able to compare those large-scale analyses of now different apicomplex and representatives as they've come online since our original CRISPR-based screening methods.
Very cool. That's very cool. So has AI changed the way you're working at all?
I think a lot about that recently. There are maybe 2 ongoing initiatives that I'm really excited about and that overlap with the extraordinary promise of AI in biomedical research. The first one is that as we've developed our method to understand gene function by doing these large-scale mutagenesis studies, we can observe each one of those disruptions as it plays out in the interaction with a parasite in living cells. This technology, which has been developed through collaborations with the Ian Cheeseman's lab at Whitehead and Paul Blaney's lab at the Broad, have allowed us to get very rich phenotypes, right, out of these perturbations that give us this grand look at all of the cell biology that's occurring in that interaction.
Even if you don't really know what the phenotype means, right? It's like a huge catalog of different phenotypes with different mutations that will allow you to sort of predict behaviors.
Precisely.
Yes.
And I think that sort of technology has been very timely to the developments in AI-based image analysis, which allows us to capture ever more subtle differences between those infected cells, between those images of infected cells, and extrapolate that to kind of comprehensive analysis of which genes must be operating together to coherently change the process of infection in a particular way.
That makes sense.
And so there, I think we have this partnership between the generation of really broad-based data that gives us sort of visuals on so many different perturbations and the algorithms that can handle that data and can help us understand common processes therein. A different area where we are trying to kind of advance experimental approaches that sort of feed into AI-based predictions is at the level of how the cell is structured. You know, as a cell biologist, that location matters, how proteins interact and come together in a living cell matters, and that many of the changes in a cell and many of the functions that we care about are not changes in the overall abundance of their parts, but in the interactions of those parts. And for a long time, a lot of our kind of broad-based analyses, if we think about RNA-seq, if we think about proteomics, have relied on measuring differences in the abundance of the parts, but not really assembling them into complex pictures. And there are new methods in proteomics, especially in the field of cross-linking mass spectrometry, that allow us to generate spatial constraints for how those parts must come together. We can do this now globally. And in fact, very recently, we just obtained the first global data sets for Toxoplasma that give us a sense of how thousands of different Toxoplasma proteins interact with one another and how the proteins themselves are structured because you're also obtaining spatial constraints for the individual parasite proteins. This, for me, is really mind-blowing.
Presumably, you might be able to use that for drug targeting of the latent infected cells, right? You know, what proteins are interacting on the surface of the parasite, how might you disrupt those interactions, etc.
Yes. The way that this can feed into AI-based structural prediction is really beautiful. I mean, I think many of us have heard about the extraordinary advances in AI-based protein structure prediction. And we are really delighted to be collaborating with the Ovchnikov lab at MIT to use these structural constraints to try to globally predict how the molecular shape of all of these parasite proteins are and how they come together. I think you're absolutely right that as we develop these methods and we're able to take essentially these snapshots of how all of the pieces come together.
Right.
We want to move towards how are they changing at
Yes.
different states, in the
Yes.
acute and the chronic state, but
Yes.
also how is the infected cell changing?
Yes.
What are the interactions that are supporting this sort of remarkable biology? And what's beautiful about that is that we can break to some extent our reliance on genetic manipulation,
Yes.
right, which has required
Right.
us to be able to do that in very specific species, and is
Yes.
difficult to
Yes.
perform in other organisms. As long as we can grow it,
Yep.
we can measure all of those molecular details,
Mhm.
which I think I'm very excited about being able to apply to other infectious diseases, including many of the infectious diseases that remain as neglected tropical
Yes.
diseases
Yes.
that have received very little attention from pharmaceutical industry, and that of course continue to cause millions of infections
Right.
around the world, and which have not benefited from all of the advances in molecular
Yes.
biology
Yes.
that have enriched cancer research, have
Mhm.
enriched cardiovascular research, brain research, of course.
Mhm.
Can we bring these tools that we almost take for granted here at
Right.
MIT and the
Right.
hub of
Right.
all of this technology,
Right.
can we bring them to those neglected tropical
Yeah.
diseases?
Very interesting. Very interesting. So you're originally from Colombia, and you spent a lot of time in your mother's genetics lab. So what was that like? How did it shape your path into science?
Yeah, I don't remember learning what a chromosome was. It was sort of so
Yeah,
so
yeah,
much a
yeah.
part of
Yeah.
our family culture. And I remember as a kid helping my mother cut out chromosomes from the karyotypes that she was assembling for her lab for cancer research or for diagnosis of congenital disorders. And this was, I think, teaching me that life, which I think is sort of so rich and observable in the natural world, came down to these invisible principles of genetic material being passed from one organism to the other, from parent to
Yes.
child.
Yes.
And so to me, that was all a little bit second nature.
Yeah.
I also grew up spending some time as of course, she would be meeting with patients or doing her work, playing around with the microscopes in
Right.
her laboratory,
Right.
observing both the cells that they were growing, the karyotypes that they were trying to make, and really learning about some of the tools of laboratory science.
It's such an exciting adventure for a kid, right? You're
Yeah.
like, wait, you can do this for a job?
I know.
Just, satisfy your curiosity, and it's
Exactly.
definitely exciting. So you made double majored in fine art, though, in cell and molecular biology. So how do you think about how your art background influences your work?
Yeah, and
how
I
you
think
about these problems.
growing up in Colombia, I sort of always had those two dueling passions, right? And I think often we see as art and science as two different poles, but in fact they're united by creativity. They're united by the desire to discover and create and share those new creations with other individuals. I think that's very central to the motivation
Right.
of a lot of scientists,
Right.
as it is to a lot of artists. And so, not being able to decide, I decided to come to the US where it was possible to
right.
undertake those dual degrees
Right.
and sort of continue fueling the two flames.
Right.
And really, as I started as an undergrad at Tulane, and over the years that followed, I really fell in love with research as I continued to do a lot of studio art. And until the very end of my undergrad, I thought I'm going to try to keep these two alive and see where things go. And it wasn't until I took the next step to go work in Berlin at the Max Planck Institute for Infection Biology that I kind of really committed to one of those avenues.
There's such an elegance to biology as well. It's sort of this aesthetic pleasure in biology that I would think you would also get from the fine arts, this
Yes.
sort of beauty.
Absolutely. And maybe the resonance between different patterns. I think that sometimes when we're overwhelmed by the facts of biology in the kind of introductory classes, it's hard for us to pick up the patterns. But the more you're in it, the more that you learn from colleagues and really kind of embrace the entire field, you start seeing the patterns of life reemerge in very distinct contexts. You start seeing convergence of design. You start seeing the reuse of common elements, and that really does enrich the picture. It gives you a sense of that natural history.
You know, it's interesting. This is a little bit of an aside, but something you saying strikes me. We have a lot of discussion about what will education look like in the age of AI, and I always think about, like, how much do you actually have to have in your own head to make those kind of connections you're talking about, rather than essentially offloading it to AI, right? That kind of pattern. I mean, AI is great at pattern recognition, but for you to see those patterns and have the kind of creative thoughts you're talking about, how do we balance that? It's going to be very interesting.
I agree. I think about that a lot when teaching cell biology in 706.
Yes.
What is the framework that we have to offer the next generation so that they know what to ask and how to think about the problem?
Exactly.
There has been this clear transformation, if I think about the history of cell biology, between a field that could be conceivably captured in a single person's mind. Right. If I think about some of my senior colleagues, Harvey Lodish, it really does seem that up to a point, he knew all the cell biology that there was to know.
Right.
And over the past 40 years, the amount of knowledge that has accumulated on distinct organisms and pathways, it really is impossible to capture in a single mind. And how do we take that leap to understanding who we should ask, what we should ask, and having a kind of global framework for how things work, and operating effectively within that framework? I think that is one of the challenges. The way that I use some of the large language models that are, of course, in all of our lives right now is really thinking about them as expanding the kind of associative memory that is sort of allowing you to say, oh yeah, I had not thought about that, and I should incorporate that.
But then you have something to incorporate it into.
Correct.
And that's the question of how you build that mental framework.
And you need an editor. You still need to be your best editor, your most critical editor. And that is the part of our brain that we often shut down when things become easier.
That's right.
And so kind of maintaining that high level of self-critique in our work, even as the large language models are making it much easier to produce things that approximate the final product.
That's right. So how did you, from all of this background, how did you find your way to MIT?
So after working in Berlin, I decided to return to the US for my PhD, which was at Washington University in St. Louis. That's where I started my work in Toxoplasma in the laboratory of David Sibley, really creative and bold scientists who would always allow us to take risks and bring in new technologies and new ideas into Toxoplasma research. And he was, I think, the one who proposed me looking into some of these fellows programs that allowed newly minted PhDs to start their own laboratories, right? Where typically in our field, we tend to do a postdoc in order to prepare for a faculty position. And so I applied to a few of these fellows programs. There aren't that many. And I got interviewed at Whitehead. And so I joined in 2012 the Whitehead as one of their fellows. And it was just an incredible experience that allowed me to pursue some of those ideas and concentrate in my research for several years. It went really well. We were able to establish some of those first genome-wide screens that I told you about. And I had the great fortune of being recruited thereafter to the biology faculty at MIT. And I really loved both the breadth and the rigor of science at MIT. It just felt like a place where bold ideas could could thrive, where you could connect on the basis of curiosity and knowledge and science with other investigators, and where their doors were constantly open to a collaboration, insight, advice. And I've really found that to be true, even though infectious disease research is a relatively small footprint of what we find at MIT. All of these interactions with other researchers working on RNA biology, working on different chemical biology approaches, collaborators in biological engineering working on mass spectrometry, have really served to enrich our research beyond belief, and have allowed us to bring into the field of infectious disease research all sorts of cutting edge approaches. And I don't know that it would have been possible, certainly not to this extent, in another environment.
You know, this was really brought home to me in the last week in two separate ways. I don't know if you've seen the documentary on Phil Sharp, Cracking the Code.
I haven't yet.
So it's on PBS. You can see it. And it really highlighted this life at MIT as this incubator of incredible science, and you know, all of the synergistic interactions between people doing different but related things. The other thing, it was yesterday was the memorial for David Baltimore, you know, the towering Nobel laureate who had such an impact on MIT, and in particular the Whitehead, founding director of the Whitehead. But what was funny was David had originally intended that Whitehead fellows would not be hired as faculty because he didn't want the fellowship to be perceived as an audition, you know. But you know, as they said at the memorial yesterday, he decided that those rules could be superseded because the people were so fantastic that there were you know, a number of the original Whitehead fellows that wound up on the MIT faculty, and you're obviously one of the later ones that wound up on the MIT faculty. So
I received the same prompt when I was hired as a Whitehead fellow.
Oh yeah, like you weren't going to be hired here.
No.
Yes.
And certainly I went through the same kind of faculty search process.
Yes.
Any other member would
Yes.
Been
that's funny. You know, MIT is a special place, and you know, I can understand the opportunity arising and grabbing it. So
yeah, and sort of trying to, I guess, retain talent and collaborations in different fields.
Exactly. So speaking of talent, finally, let me just ask you this. You know, you've been open about being gay, you've been interested in support of the LGBTQ community. From your perspective, what steps could universities in general, and MIT in particular perhaps, do to really support people from diverse backgrounds? Because we are about getting the very best talent from all quarters, and we want to make sure that everyone's here feels like MIT is their home.
Absolutely. I think openness and inclusivity are challenging things to implement, right? And I think that we need to have those conversations that open our eyes to different perspectives in order to be able to recognize differences that otherwise go unnoticed to us because of the limits of our own perspectives. And so for me, it's just been so enriching to be able to listen to some of those personal stories from Dean Mavalvala, from other colleagues throughout MIT. And it is, I think, in sharing that complex process of learning how to belong, of feeling like sometimes we don't belong, that we end up finding, I guess, a common framework for our distinct humanities, right? And so I do think that having opportunities to share that go beyond our professional persona and to listen to each other's stories in an open fashion, I think it's those moments that, without knowing it, help other members of our community know that they belong.
Yes.
And so for me, I think in my own professional development, I have felt at times vulnerable based on my sexual identity, at times based on my immigration status, based on my ethnicity, there are moments where I feel like I don't belong, or maybe I belong for the wrong reasons, right? I remember when I was hired to the MIT faculty, one of the graduate students, and I wasn't much older than they were, so we were all kind of friends. One of the graduate students at Whitehead, actually someone from Latin America, said, "Congratulations, I wanted to be the first diversity hire."
Oh, wow.
And it was actually the first time that I thought, oh wait, is that why they hired me?
Yeah.
And it's such a pernicious thought, right? And
yes.
And I mean, I think now I have the confidence in what we have achieved,
right?
In all of the wonderful research that I've been able to lead by so many talented researchers, not to take undue credit, that I know that was a really misdirected statement.
Yes.
But I think that we are all susceptible to those moments where we wonder what role our identity has in the achievements that we've garnered. And I think knowing that others have similarly felt uneasy about their situation and doubted themselves at different times sort of helps us put those experiences in context.
Right.
And recognize that belonging is an ongoing process, right? And that when we feel strong, when we feel powerful, it is incumbent on us to open those doors for others,
right?
To tell those stories, and knowing that not everybody feels equally comfortable and on equal footing to do that.
That's right. I mean, when we hire for the very best talent, we're going to have a diverse talent pool. Doesn't mean that they're a diversity hire. We've hired the best talent.
Correct.
And people are going to come from a wide range of backgrounds. And the thing that's really struck me, I've now been at MIT for 3 years, the thing that really strikes me is the sort of people are accepted, all kinds of people. You know what I mean? It's just a very interesting, diverse community from, maybe people who are very conventional in their viewpoint to very quirky, from very left-wing to very right-wing. I mean, it's just been, incredible, because I think the bottom line is everybody here wants to do their best work. And that's the ultimate focus.
Absolutely. I think the science and the achievement. But I also think that process is a human process.
Yes.
Right?
That's
right. And so we actually cannot dissociate those two.
No, that's right.
And so how do we create a community where we recognize and accept each other's differences and each other's humanity? That also requires a certain proactive approach towards having difficult conversations.
That's right.
Having civil and respectful conversations. Right. And in that process of disagreement, not forgetting each other's humanity, right?
Well, that's right.
And all of the things that we actually share.
That's right. And this is a much longer conversation because this is a problem well beyond MIT. But I'm glad that we got to have some of this conversation today. I learned a lot, both about parasites and about you. So it's been great. And to our audience, I want to thank them for listening to Curiosity Unbounded. I very much hope you all will join us again. I'm Sally Kornbluth. Stay curious.
Thank you, Sally.